All right, right on. So, hello everyone. My name is Sky Lee. I'm the Associate Director at the Genomics Core at University of Colorado Anschutz Medical Campus, and I'm really excited to talk about our experience with the T7 sequencer. We have one at our core, and we've had them for about a year now, and very happy with it. And namely, whole genome sequencing is not something that we've been doing a lot at our core, but thanks to the T7, it's something that we've been able to enable. And I'm here to tell you all about our experience with it, specifically the data that we generated, but more on the QC side of things. So it added a lot of credibility to the two talks that came before me. So without further ado, let's get into it. So before we get into it, let's talk a little bit about whole genome sequencing. Why should we perform whole genome sequencing? Well, incidentally, WGS is the most powerful tool for testing genetic disorders such as mutations that cause diseases, some forms of cancer, and tracking infectious disease outbreaks. And importantly, whole genome sequencing encompasses not just the coding region, which is the exons, but also the non-coding regions, the introns. So whole genome sequencing will provide you with a comprehensive map of a person's entire genetic makeup. And you might be asking yourself, if whole genome sequencing is so great, why doesn't everybody do it? Well, the answer, is pretty much cost. It's really expensive. And even if we just kind of rewind the clock back 20 years ago, it would have cost $100 million to sequence a human genome. And even into the recent past, if you asked us, hey, can you do whole genome sequencing a couple of years ago, we would have said close to $1,000. But thanks to the T7, we can actually do it for only $350 per sample. And this is all included with respect to reagents and labor and all that. And just to kind of put into perspective, whole exome sequencing is also $350 at our core, but compared to whole genome sequencing, you only get 1% of the data, okay? Because you're only looking at the exons. So pretty remarkable stuff. Our approach was specifically looking at the same 20 samples and using two different preps. And you might be asking why 20 samples? Well, incidentally, just due to the T7's flow cell configuration, we have to do 20 samples if we're talking about 30x coverage. Specifically, it does 6 billion reads per flow cell, and 30x coverage, that's roughly 300 million reads per sample. So 20 samples perfectly fits into an entire flow cell. And we took two approaches, the PCR-free and the PCR'd approach. With the PCR-free, it's actually a new kit for us. So we have no experience with this, and this is a native kit offered by Complete Genomics. And some pros and cons associated with it. Mostly here, it's just the cons, but in terms of the data, it's going to be mostly pros. But since it skips the PCR step altogether, we require a lot more DNA to start with. And just the way that the kit is configured, which is enzymatic digestion, the DNA integrity has to be really, really good. Now, you contrast that with the PCR prep, which is something that we've been doing it for close to a decade at our core. That specific kit is made by NuGen TCAN, and these are actually Illumina-ready libraries. So we use this for our NovaSeq sequencing, but with the T7, you can actually use a conversion kit to take Illumina libraries and sequence in the T7. Specifically, since it does incorporate PCR into the steps, we only need 100 nanograms. And of course, we don't use the full 100, but that's what we asked for. And even degraded DNA is okay because specifically with this kit, we do mechanical fragmentation, Kovar shearing. And when we started digging into the data, we were quite impressed. So just starting with the Q30 scores, what exactly is Q30? Well, it measures the probability that a base call is called incorrectly, and specifically Q30 means 99.9% of the base call is accurate. And this is not just with respect to Complete Genomics and T7. Q30 is the benchmark for quality in all of next-gen sequencing. And when it comes to Illumina runs, you can kind of expect to generate between 70 to 80%. At our core, maybe more like 85 to 90%. But with the T7, we were kind of really impressed by the fact that for both preps, PCR-free and the PCR data set, it was generating Q30 scores of between 94 to 99%. And I mean, 99%, that's pretty remarkable. So as far as that goes, fantastic. Duplication rate, this is something that we also looked into. Duplication rate specifically looks at the fraction of mapped reads marked as a duplicate read in a particular data set. And it's actually not very useful just because, as the name implies, if it's duplicated, there's going to be no additional information from the secondary, third read, et cetera. So duplicate reads offer no additional information and are bioinformatically removed during a process known as deduplication. And unsurprisingly, the PCR prep did exhibit duplication at a rate of 7 to 14%. And of course, that makes sense, right? The PCR step, it's going to duplicate your DNA, right? Whereas the PCR-free prep exhibited around 1% duplication rate. So yeah, so all of that is consistent so far. We also looked at uniformity. Uniformity is another very important metric that you want to look for. That describes the read distribution along the target regions of the genome. So specifically, uniform coverage reduces the amount of sequencing required to reach a sufficient depth of coverage for all regions of interest. And again, if we kind of I said like 30x coverage is around 300 million reads, right? Can you imagine if, we amplify the same region of the genome and sequence it 300 million times and give you the data, and, you'd be rightfully pissed. You want to have some uniformity, right, across the entire genome. So that's a very important metric. And sure enough, by following these preps and sequencing, both the PCR-free and the PCR prep exhibited well over 96% uniformity. So far, so good, yeah. Obviously, when we sequence, we're aiming for 30x coverage, so naturally, we want to look for depth. Did we actually, in fact, achieve 30x coverage? And the short answer is yes. And here, that's a little bit more background. 30x coverage for the whole human genome translates to 300 million reads, 600 million paired end reads. And what we saw was that with the PCR-free data set, it's a range between 20x to 40x coverage. So I guess on average, 30x. Some samples got more, some samples got less, but yeah, pretty close to 30x. The PCR data set was exhibiting between 20 to 30x coverage, whereas for some odd reason, those 4 samples were exhibiting 40 to 60x coverage. I don't have a good understanding why that happened, but the data's the data, so there it is. So both the PCR-free and the PCR prep are capable of generating 30x coverage on average. Great. We actually dug in a little bit deeper just to see what it looked like looking at the coverage from low, medium, high, starting with 1x. I mean, that's the bare minimum. If we can't even do this, then what are we even doing, right? So what fraction of the genome can be mapped with at least 1x coverage? And unsurprisingly, pretty much most. Both PCR-free and the PCR data set mapped with well over 98.2% coverage, with at least 1x coverage, meaning you're going to have at least one piece of DNA, a fragment of DNA that's going to map onto the genome at least once, okay, for 98.2% of the genome. Fantastic. What about 4x coverage? So this is a little bit more the medium, maybe lowish medium. And so we asked that question, what fraction of the genome can be mapped at least 4x coverage? And yeah, so both for the PCR-free and the PCR data set, it's mapping above 96% at 4x coverage, which is great. So what about the absolute tippy top? 30x is what we're aiming for on average. So what about as far as like the end of the error bar, basically? How much of the genome is mapped at 30x coverage? Well, that's a little bit more variable, where both the PCR-free and the PCR dataset had a very wide range between 5 to 80% mapping at 30x coverage. Yeah, I guess that's fine. Like some of these samples, I guess like some of the regions will exhibit 70% of the genome. For some of these samples will have 30x coverage, whereas other cases only 5% of the genome will have 30x coverage. But I suspect if we start bringing it down to 29, 28, 27, we're going to get quickly to sufficient parts of the genome being covered. But again, the data is the data, so there it is. And I think that this is a good kind of segue briefly into why coverage is so important. So we talked about 1x, 4x, 30x coverage. Well, what you're seeing here is just sort of an example of 11x coverage, where you can see here there's the reference genome, that's the GRCH38. And just to kind of briefly talk about that reference genome, this is a very, very comprehensive benchmark. Everybody around the world is contributing to this reference genome, and this is version 38. So maybe in a couple of years from now, you'll be seeing GRCH39, and that'll be the new reference genome that you'll be aligning your data set to. And again, coverage is so important because it can really eliminate outliers, if that makes sense. So specifically, if you look on the right here, you have 11x coverage, and you got some of these bases that were, for example, over here, it's clearly it's an A, whereas you see A once, but you see C 10 times, right? Or here in this spot, you see G once, but you see T 10 times. So these are ways where bioinformatically you can identify whether or not something is due to a PCR error, sequencing error, et cetera, versus a bona fide SNP, single nucleotide polymorphism. You're going to see it in a particular spot, and not just in the exact same spot, but specifically the mutation is consistent as well. So the G is turning into an A rather than G into a C or a T. So those are going to be more or less non-random. And this is a very important reason why coverage is so important. The more coverage you have, the more confidence you can have in the data when you identify particular nucleotide polymorphism or variation. So yeah, going back to the GRCH, it is a very comprehensive benchmark, and any variants discovered, SNP, INDEL, structural variants, et cetera, in your whole genome sequencing data set, if it aligns to that reference genome, that provides further confidence. So in many ways, I like to think about it like a positive control. Like maybe you're the first person to ever discover a particular mutation, but realistically, you're competing against the entire world who may probably have seen that same mutation elsewhere. But if you make 2 independent discoveries, maybe without communicating with each other from across the world, and you're both observing it, that's actually, I think that's a positive thing. That tells me that it's probably a real observation that you're seeing. So So kind of digging back into the data, the 20 samples that we did for the PCR-free and PCR data set, up to this point, this is really the kind of the quality metrics associated with the T7, but we also wanted to kind of dig into the data a little bit just to see what that looks like. And with respect to this, the tools that we used was GATK 4.0 haplotype caller, the single nucleotide polymorphism database, DBSNP, and RTG tool for variant calling comparisons. So total SNPs identified. So an SNP is a single nucleotide change that is seen in more than 1% of the members of the population. But if you see it less than 1%, that's called an SNV, single nucleotide variant. And the conclusion is that for both cases, the total SNPs detected for the both PCR-free and the PCR dataset are highly consistent. So great, at least between the kits, we're not seeing much variation so far. Novel SNPs, but this is where it gets a little bit more interesting. A novel SNP may be, and I emphasize the word may be a unique SNP discovery in the data set. So a novel SNP can be determined by comparing your whole genome sequencing data set of those SNPs and then aligning it to the reference genome. And those SNPs will be deposited in the DBSNP. And again, this is what I'm referring to. So according to the data set, it's saying it's identified about 2.5% of the SNPs are considered novel, right? And it's 2.5% for the PCR-free data set, but actually for the PCR data set, it's about 0.5% higher. So there's a bit of a discrepancy because they're the same 20 samples that got sequenced. So this is where I was kind of alluding to earlier. From the sample prep point of view, the PCR-free is going to be harder because you need to get more DNA to start with, but as a consequence of skipping the PCR, you reduce the possibility of PCR errors being introduced to your data set. So 0.5% doesn't sound like a lot, but we're talking about 3 billion bases. 0.5% of that is 15 million bases of just noise that you're gonna have to contend with. So during the bioinformatics step, you're gonna have more challenges. So it's a matter of what your trade-off is ultimately gonna be. But yeah, I mean, another interpretation that I'll add to this is if 97.5% of your data is aligning to the reference genome, then again, that's saying there's a lot of confidence in your data set because it's aligning with what other people have observed around the world as well. So with that said, I will say the caveat is that whatever novel SNP that you discover, it could be real, but it could be an inherent error or both. So really, I don't think you should get too excited about it, but this is a place to start digging into if you are indeed looking for novel SNPs. We also look for indels. Indels, obviously, as the name implies, insertions or deletion of bases in the genome. And it's a little bit trickier here as well. The PCR-free data set exhibited 96% alignment to the reference genome, which is excellent. But with the PCR data set, it's aligning at 75 to 85% rate, right? And again, I'll remind you, these are the same 20 samples that were used with these different library preps. So there's more discrepancies. This sounds like a challenge bioinformatically. Very real likely what happened was that the PCR steps, like I said, introduces PCR errors, but during these indels, when you align them, I believe they were maybe a little bit too stringent, and whenever they saw any base pair mismatch, which in the particular region that it's aligning to, it just threw it away. So that's why the rate must be much lower. But if you forgave some base pair mismatches here and there, maybe the alignment would be better. So that's more on the bioinformatics side of things. But once again, just kind of pointing out the fact that you're going to have to overcome these challenges during the bioinformatics step if you do offer the PCR prep. So yeah, the PCR-free dataset has more concordance with the reference database, therefore much more confidence in the indels that you did identify, and the PCR dataset exhibited more false negatives. TITV ratio, this is a fun little metric that we looked at. This looks for the ratio between transition and transversion. And you can see in the upper right-hand corner, just by the sheer virtue of looking at the number of arrows, there's more possibility for transversion. So 1, 2, 3, 4 versus transition in the blue arrows, 1 and 2. So if the mutations were totally random, right, the TITV ratio will be 0.5, just 2/4. However, in reality and nature, it's not random. The transition mutations happen much more greater frequency. Just kind of one example off the top of my head is cytosine deamination, where let's say this amine group is replaced with an oxygen, it becomes a uracil, and there you go. And then that gets interpreted as thymine. These kind of mutations happen in biology. So, so if you look at the data, if it's good, then for whole genome sequencing, the TITV ratio should be around 2. And if it's above 2, close to 3, then that implies that the data set is highly biased. Whereas if it's close to 1, then getting closer to 0.5, like I said, the data set is exhibiting a lot of high error rates. And lo and behold, we're very happy with this too. The TITV ratio is close to 2.0 for both data sets, indicating high confidence in the data. So fantastic. Of course, like up to this point, we're looking at the different polymorphisms, et cetera, but are they actually correlated between these 20 samples for the PCR-free and the PCR data set? And we use this tool called RTG VCF evaluation, variant calling. What this will do is perform sample-to-sample comparison. So sample 1 from PCR-free versus sample 1 from PCR, and sample 2 with sample 2, et cetera. And specifically what we looked for is precision and sensitivity, where precision looks for false positives and sensitivity looks for false negatives. And for obvious reasons, we chose the PCR-free data set to be the reference that we compare to the PCR compared to the PCR-free data set. And yeah, I mean, it was pretty good. For the most part, the PCR data set exhibited 90 to 95% agreement with the PCR-free data set, meaning one mutation that you see in the PCR data set, there's a 90 to 95% chance you're going to see it if you opted for the PCR-free data set library prep, except for these two samples, outliers, sample number 17 and 19, which exhibited 85% agreement, which, you know, it's still not bad, but yeah, I don't have good explanation for what any of these why it's happening, but the data is the data, so there it is. So in conclusion, what we can tell is that the PCR-free prep yields relatively, and I italicize relatively because it's relative to the other prep, higher quality data, but the caveat is it requires 500 nanograms of genomic DNA with good DNA integrity, where you contrast with the PCR prep, it exhibits relatively lower quality data, but you only need 100 nanograms of genomic DNA and degraded DNA is okay. But you know, those are somewhat relative subjective standards between these two preps. On an objective level, both preps generate high quality data that exceeds QC benchmarks. So specifically, you know, in the field, you know, if you read a manuscript or a paper, the vast majority of people are going to be doing the PCR prep, PCR free, just because that barrier to entry with respect to the sample is high. I think people just naturally go for the PCR prep. And incidentally, like for example, if you have FFP samples in biobanks and archives, then obviously the PCR prep is going to be a lot easier as well. But you know what that tells you is that if you offer the PCR data set prep, then you're getting basically the same data quality that is already out there as the current paradigm. So no worries there. But like I said, thanks to the T7, you can do it at a fraction of the cost that it would have been done traditionally. The PCR-free data set, you get better data quality than what's out there, but you still get that insanely low cost. So for only $350 per sample, at least at our core, this assay is an invaluable tool for first pass mutation screening, DNA damage experiments, GWAS, genotyping cell lines or genotyping mice strains, and more. But I'll once again add another caveat and reminder, just in case I've done too good of a job, explaining all this stuff. The whole genome sequencing is still a bulk sequencing experiment, meaning it's really not like single cell or spatial, where it's more like the centerpiece of your project or manuscript. This is more like if you have an overt phenotype and you don't know what the underlying mechanism is. This is a great tool just to kind of get an understanding of what may be of causing the mechanism that you're seeing, the phenotype that you're observing. So this should really lead into the further experiments, as in like it's kind of refining your hypothesis for maybe that single cell or spatial experiment. But even then, like here's some easier follow-up experiments that you can do to make sure that your observation for whole genome sequencing is correct. You can do knock-in, knockout, CRISPR mutation experiments in cell lines followed by bulk RNA sequencing. You can do amplicon-seq. If you identify a very interesting mutation, you can take that same DNA sample and just amplify that specific region and just double check if that mutation is really there. Or if there's a codon change in that particular site, that a nucleotide, and that leads to a change in the amino acid, maybe you can subject to mass spec and find out if you're seeing that mutation translating, no pun intended, at the protein level. And I'm sure there's more that you can kind of think of on your own too, but those are some 3 simple examples. So with that said, that's all I've got. I really appreciate giving me the opportunity to talk about our experience with the T7, and yeah, I'd be happy to take questions over here with the 2 gentlemen.